Hello guys and welcome back to Excel Academy. I'm Dr. Ruby Joshi and here I am with another questions. Actually guys, this is a part of the previous question itself. So now I just got this question, so I just thought, why not making another video on this? Okay, so actually it is related to the previous question itself. So if you remember in the previous question, I have talked about the incorporation assay, right? And this assay actually measures the DNA which is synthesized using the labeled DNTPs. Okay, so in the previous question we talked about like how to separate them and why there is no radioactivity if beta and gamma are used, etc., etc. Okay, now this is the third question and it is very simple. Like if you know how to design an experiment, so whenever you design an experiment you always keep there's something called control, right? So using that control only you are going to carry out the experiment, because you need a reference with which you can compare your data. So we have got the different types of controls, like we have got the negative control, ठीक है, एंड वी ऑल्सो हैव द पॉजिटिव कंट्रोल. अब इनका क्या मतलब होता है? नेगेटिव कंट्रोल मतलब वहां पे आपको कोई भी रिएक्शन प्रोडक्ट नहीं मिलना चाहिए. पॉजिटिव कंट्रोल मीन्स यू शुड गेट. अब इसके बीच में लाइज योर सैंपल, ठीक है, जिसकी एक्टिविटी वी डोंट नो अबाउट इट. So in the same way, in this case, in the incorporation essay, so the question was asked regarding the filter binding. So if you go through the lecture video, you will see that we have talked about the filter binding in order to separate the unincorporated and the newly synthesized DNA. Okay? So over there, now we are asking the negative controls that would ensure that your filter is separating the unincorporated labeled DNTPs from the DNA. ठीक है, तो अभी हम उसमें पूछ रहे हैं कि नेगेटिव कंट्रोल आप क्या यूज कर सकते हैं. अगर आपको ऐसे मालूम है देन यू कैन रियली इजीली गिव द आंसर. नहीं भी मालूम है देन ऑल्सो यू कैन एक्चुअली थिंक अबाउट इट. ठीक है, तो इसमें आप क्या कर सकते हो? For the control, see, as I said that negative control means there has to be no reaction. So in this case what you can do is that you can run the same DNA synthesis assay, but you can carry out in the absence of the DNA polymerase. So what will happen if there is no DNA polymerase? Just don't add DNA polymerase as your negative control. So what will happen if there is a no DNA polymerase? Then there will be no new synthesis of DNA, correct? So only the primer template junction will be left and it will not be labeled. Okay. Now, if you properly filter the reaction, which is having this primer template junction as well as the labeled DNTPs over a charged membrane, right? We need a charged filter binding membrane because your DNA is going to be negatively charged and hence it is going to bind. Okay, so that's why we are using the positively charged membrane. So what we are going to find that the radioactivity is not seen. क्यों? क्योंकि आपका प्राइमर टेम्पलेट जंक्शन भी बाइंड नहीं करेगा और लेबल डीएनटीपीस भी बाइंड नहीं, अनइनकॉर्पोरेटेड लेबल डीएनएस भी वहां पे वो बाइंड नहीं करेंगे. ठीक है, सो नाउ यू कैन कंपेयर दिस to the same reaction which is having the DNA polymerase. Okay, now if you're worried about any possible effects like the label DNTP is binding to DNA polymerase, then you can actually give the protease treatment also. अगर आपको लगता है कि before doing the before carrying out the experiment अगर ऐसा कुछ हो रहा है, so you can have the protease treatment. Okay, so guys, I hope you got the idea over here कि negative control कैसे use करना चाहिए. ठीक है, of course you cannot have the absence of the प्राइमर जंक्शन टेम्पलेट. आप ये नहीं कह सकते हम उसको हटा देते हैं. ठीक है, यू नीड, बिकॉज आपको देखना है कि उसमें इनकॉर्पोरेशन हो रहा है या नहीं हो रहा है ऑफ न्यूली सिंथेसाइज ऑफ न्यू डीएनटीपीस और नॉट. So that's why we remove the DNA polymerase. So DNA polymerase नहीं रहेगा, so what will happen? No new synthesis will be there. So अब हम इसमें हम पता कर सकते हैं that whether it is the filter is able to separate the newly synthesized DNA or the unincorporated 32 label DNTPs or not. ठीक है, थोड़ा सा कॉम्प्लिकेटेड रहा होगा, बट लिसन प्रॉपरली एंड थिंक अबाउट इट, यू विल गेट द आंसर. ठीक है, सो दैट्स ऑल फॉर टुडे and I might be soon starting the new series also, so keep watching guys, it is going to help for your exams. Thank you.